We can see it.
Okay, cool. So thanks, Tamsin. Yeah, hi, I'm Ellie. I think I recognise some faces maybe that took part in this project, so it's nice to see you. Yeah, I'm a postdoc, not a postdoc, I'm a PhD student at the CBCD, and together with Rianne, who is a postdoc, we're going to talk to you about the Braintools project. So some pictures of us there wearing the Braintools EEG kit, just to show that, you know, we do it too at times. Ooh, sorry, just, okay, whoops. Sorry, it's being slow. Okay, there we go. So the Braintools project is a global project which aims to develop a toolkit for measuring children's brain development. So this project is actually across 3 sites, kind of worldwide, here in the UK, in the Baby Lab, in the Gambia, and in Delhi in India. So some of our colleagues have been out to the Gambia and India, thankfully pre-COVID, and gathered data out there. And myself and another colleague, Teresa, kind of collected data here in the UK. So yeah, if you took part, you'll probably recognise me from that. So yeah, and there's some pictures there from our actual participants that we had. So today I'll just be talking about the UK part of this project. A similar thing was done at the other locations, but kind of data is in the process of being analysed from those locations. And today we're just going to focus on the UK version part. So why is this study kind of helpful and important? To me, this kind of comes under 3 different questions. So firstly, why are we interested in brain development? Secondly, kind of why does it need to be global? And third, kind of what's the need for a toolkit? What's that all about? So why brain development? Well, we know kind of from previous research that there's evidence that we might see developmental differences in the brain and in brain activity kind of at a younger age than what we see behavioural differences. So kind of some of this works from children who have autism. So for instance, behavioural differences might not kind of become apparent till children are kind of toddlers at earliest, preschoolers, but actually there might be brain kind of differences in their brain activity during infancy. And so that might be quite helpful in being able to identify these children from a much earlier age, so we can put support in place. That being said, there's still a lot we don't know about the links between kind of these brain differences and these behavioural differences. So there's still a lot to find out. This is there's a lot that we don't know yet as well. But yeah, I guess kind of the aim, like I've said, just to reiterate that if we can kind of understand what's happening in brain development and how that might be leading to some children having kind of optimal outcomes and some having less than optimal outcomes, and maybe we can understand how to support those children better. So that's kind of why brain development. So why global? Well, there's huge variability across the world in access to kind of health services and clinical services. And particularly in lower income countries, kind of clinical services, so for diagnoses and things, is often very much lacking. And so with the goal of being able to develop a tool which can help find children who are in kind of need of support and might have poor outcomes, kind of if we can do this globally and kind of find a tool, find a way of doing that is less kind of relies less on clinicians and is less time consuming, then that might be really helpful. As a secondary kind of, not a secondary aim, we know that kind of environment has a huge impact on children's development and brain development. And so if we can kind of look across cultures at how kind of different environmental factors might be affecting brain development, that could also be really interesting and helpful. So that's why global. Why a toolkit? So all these outcomes, all of these aims and things, cool things that we could do are great and really interesting. But first we need to be able to know we can trust our measures. So kind of there's technical advancements that mean there's these, as we'll talk to you about today, kind of more portable and wireless systems that we can use, which kind of create opportunity for doing studies in kind of field sites, so across the world and across the UK. But we actually need to know, that we can trust them. And as yet, they're still quite new and not that studied. So kind of here, we're looking, are they reliable? So there was also a few other differences in our protocol that we kind of need to check. Are these an effective way? Are these working successfully? So we first need to check how feasible our protocol is. So that's, is it doable? Can we do this with this age children in these settings? Is it going to work? And then how reliable? So can we trust what we're doing? So what did we do? Well, we invited 2.5 to 4-year-olds into the Braithwaite baby lab for 2 visits, which were kind of a couple of weeks apart. And that 2 visits is really important because that's how we assess the reliability. So basically what we're looking at is whether we get the same kind of response, kind of brain response at visit 1 as we do at visit 2. And that will tell us if it's reliable. If those are completely different, then it's probably not a reliable measure because we're measuring totally different things at different times. But if it's consistently we're measuring the same thing, that's an indicator that it's quite a reliable setup. So as I mentioned, we used a brain measure which is called EEG, which I think Rianne will talk to you a bit more about in a little bit, but basically it measures the kind of natural electrical signal that our brains give off whilst we do things. And we also used simultaneous eye tracking, and that's kind of one of the other differences that we used in our protocol. So we kind of weren't really interested in which part of the screen children were looking at, but we used eye tracking as a way of presenting stimuli. So instead of kind of just showing image after image after image, regardless of where the child was looking, we used eye tracking to pick up when the child was looking at the screen and then presented the next image. So we can have kind of more trust in that the children are actually looking at the screen when we think they're looking at the screen. And also it kind of allows the child to pace it themselves a bit more, kind of hopefully. Well, what did we do? So children sat on, children sat with their parents, usually on their knee, and kind of watched the screen. And there are kind of 2 elements to the study. So there's a visual part and an auditory part. So in the auditory part, children heard tones of different pitch and kind of watched bubbles or a silent video. And in the visual part, they saw kind of mixed up static images and videos. And today it's the visual part and specifically the static images, which we're kind of interested in and going to talk to you about. So if you can see along the bottom here, you can see lots of different kind of images of faces and animals and checkerboards. These are kind of the stimuli that we're interested in. And again, Rianne will talk to you a bit more about this in a little bit, but there's kind of, from previous research, there's expected brain responses that we kind of expect to see in response to these images. And you'll notice that some of them are upside down and some of them are the right way up. And again, this is because there's kind of different brain responses that we expect to see in response to upright and inverted images. And just above that, you'll see, it's kind of small, sorry, but that's kind of what the children saw. So it was a dark grey screen with just a single image on it. And the first one at the left-hand side with the little duck, I don't know if you can see my mouse actually, but that's kind of what we call an attention getter. So children kind of saw that in the middle, like a fixation thing, and when the eye tracker detected that children were looking at that, it presented the next image. So in this case, it was a face. And I think I'll stop there. Rianne's just I'll pass over to Rianne now, who is going to kind of show you a bit more about the actual EEG setup and the system that we used.
Okay, I'll just share my screen. Can everyone see this? Yeah. Okay. So I'll tell you a bit more about EEG and what we actually did with the data and what it tells us. So what do? We use an EEG system, and this is a portable in-audio system. So in a lot of previous EEG system, there's this big cable that measures the signal and then puts it through to the computer to measure it. But this system is actually wireless. And I also have a cap here. I don't know if you can see me, or maybe I should stop sharing my screen. What do you think, Ellie?
We can see you.
You can see me? Okay, great. So this is like one of the caps, and it's just neoprene, it's some sort of swimming cap, what we call it. And there's all these holes in there that we use to measure the brain, actually. So we have these metally bits here, and then we have the brain sensors, whoops, or our EEG sensors, which look like this. And these we then click onto the metally bits, like here, like so. And then we use 8 different sensors of these, which we place all kind of across the cap. And then at the same time, you may also see, not in this picture, but in the previous pictures, all the toddlers were kind of having some sort of ear clip on their ear, which is this one. And we're using this one as a reference because we're always measuring the EEG response relative to something. So in this case, we're measuring it relative to the ear. Similar, for instance, like you would be measuring the height of a mountain relative to sea level. In this case, we're measuring brain responses relative to an ear, which sounds a bit weird, but that's how we do it. And then, as Ellie was mentioning, we're not only measuring the EEG, the brain signal, but we're also measuring whether the child is looking at the screen or not. And we asked all the parents to wear these funky eyeglasses to make sure that we pick up the actual eyes of the kids and not the actual eyes of the parents, because that might also happen sometimes. So then this is what the setup looks like. If you look at the slides again, this is Georgia, who run the Braintools project in India. And there was a kid looking at the screen, and under here is an eye tracker, which measures whether the kid is looking at the screen or not. And then we only present the stimuli when the kid is looking. In previous studies, we would just be showing the stimuli, and then later on, we would take out any stimuli when the kid was not looking. So in this case, we're kind of already controlling for that when we are collecting the data. Let's see, next slide. Okay, so I just want to tell you what we're actually measuring with the EEG. The EEG measures the synchronized activity of groups of brain cells. So you have lots of these brain cells, neurons, in your brain, about 100 billion. And when there are groups of these that are all firing at exactly the same time, the signal is strong enough that we can measure it at the top of your head. So here, so this is the cortex where all those brain cells are. And then we can measure the signal that travels through the scalp onto your EEG sensor. And then we see a signal like this. So it just goes up and down. And we can measure that for all the different electrodes that we're using. So we have these different electrodes. The green ones are all the possible electrodes, but in this study, we only used the black ones. Then you can put your electrodes across any region that you're interested in. And in this case, for the Braintool project, we were really interested in visual processing. And we know that visual processing happens in the occipital lobe. So we were also very interested in these electrodes at the back. Cool. So then during the recording, this is what Ellie and Teresa did. Once they put all the caps on the kids, and they started the protocol. And then when the protocol was going on and the kids were looking at the screen, this is kind of what they were seeing on the screen. So each line, each trace is one EEG sensor. And here you can see this one is the most frontal one. And we can see that there are all different traces for all the different sensors. So there are 8 sensors in total. And actually what's very interesting is that any muscle noise is much stronger than your brain noise. So if you see, if there's anyone who is blinking, which is like a muscle response, then you would see that in the EEG. and you would see a stronger response frontally. So here, this kind of peak is actually a blink when the kid was blinking, like doing the eyes. And you can see it again here. So this is actually looking very nice. This is beautiful EEG data. If we can see the blinks, that means that it's very nice. And this kid was sitting pretty still during this. So this is what we're looking at. And then we can further analyze the data. And this is then going back to what Ellie was talking about. We're very interested in how the brain responses are locked to events. So in this case, whenever a face is showing on the screen, which is an event of a face, we're then looking at the brain response immediately after that. And that's what we call event-related potentials. So in this case, we've been looking at the responses to the static images, to the faces, the animals, and the checkerboards. And then the processed data, after we've done the pre-processing, kind of looks like this. So there are specific shapes of the EEG that we're looking at, where the trace on like the back of the electrodes at the back of the head just go kind of up and down. And then we look at different components. For instance, this is a P1. It's just terminology that we use. And we know that this peak here, for instance, is longer when there's a stronger contrast. So that's why we're looking at the checkerboards. And then we also have different traces that we're looking at for the faces. For instance, an N290, which is related to face processing, and a P400. So the P is the P1 is called the P1 because it's a positive peak, and the N290 is called the N290 because it's a negative peak. And then the P1 is a positive peak again. So that's what we're looking at. And then when we look at, so yeah, that's the analysis that we did. But what we were very interested in this study was whether this paradigm was actually feasible, because we're now trying to combine both the eye tracking during the data collection and the EEG, which hasn't really been done before. So we just wanted to know if kids would be able to look at this, or whether they didn't actually like this. So we recruited many kids, or actually Ellie and Teresa did. They asked 61 kids to come, and actually out of these kids, we were able to get some data from 52 kids. And then if we would think how much data we would need from each kid to include in our study, we looked how many kids we would be able to include in the study. And actually it was a lot. It was like about 90% or 95%. Whereas in other studies where we just show the stimuli without actually using the eye tracking and the gaze control, so the kids can face it themselves, this percentage would actually be much lower. It might be like 75%, because we have to take out all the trials when the kids are not looking. So actually this shows that our paradigm is feasible, and that the kids were looking at our task, which is very good. So next, we also wanted to know whether our responses were actually reliable, that if we measure them at one time and if we measure them 1 or 2 weeks later, that it would still be very similar. And this is actually very important when we're looking at development, because you want reliable measures. If you want to measure something in infancy and want to use that to predict later behavior, then we need very reliable measures that we can trust upon. So in this case, at time 1, for instance, we're looking at how long or how big this peak is, and also at what time, which we call latency. And then we do the same thing at time 2. And then we're looking at how similar these measures are. And this is what we found. So here we have the different measures. The P1 latency and P1 amplitude is how the latency is how late the peak was, the amplitude how big the peak was. They were actually fairly or moderately reliable, as was the N290 amplitude. But some other measures, like the N290 latency, was not that reliable, so it's probably very variable across time in these kids. And then we had this other measure, the P400, which was actually very reliable. So we kind of see that on average, our measures that we use are moderately reliable. Some are better than others. So that information we can use in future studies to see what kind of measures we should focus on. So to conclude, the Braintools paradigm that we've been using with both combined EEG and eye tracking would allow us to include more participants, and also all the kids were looking through the protocol throughout the session. So actually it's a very feasible paradigm, which is good to know. And also our brain measures were very moderately reliable across sessions. So this suggests that there is a potential use for global studies in examining early brain development, and then how this relates to later behavior. And that was it.
Brilliant. Thanks so much, you guys. Ellie and Rianne, that was really interesting, very informative. Does anyone have any questions? Rianne, if you stop sharing, then we can...
I'm trying, how do I...
Where it says share screen, mine's in the bottom. Can you do a stop share?
I don't see it in the bottom.
Mine just had a floating thing that just said like stop share, just somewhere on my screen, but I don't know.
I see the participants. No. Oh wait, it's on my top. I see it.
Oh, okay. All right. Well, thank you guys. Questions? Does anyone have anything they'd like to ask? I can see some of you joined because I think your children. Oh, Kitty, yeah, go for it.
Can I ask what is probably a really stupid question? What makes it a negative peak, a negative response? We're in the as it peaks and troughs.
Yeah.
What makes it a negative response? Is it that what makes that?
So it's kind of just goes up and down anyway, and then we're using a baseline. So just before the event, we have about 100 milliseconds that we then baseline it. So we're kind of moving the whole thing up and down, and then some of the peaks are going to be below that axis. So then it's going to become negative. And there's also variability in that. There's a lot of individual reliability. So some people might have a more negative peak, and some people might still have a peak that goes down, but then it might be positive. So it might be above that axis. If that makes sense.
I think so. Thank you.
Definitely not a stupid question.
No, definitely not. There are no stupid questions.
Hi.
Hi.
Sorry, again, this was really interesting. I'm so glad to see the project going well. I was just wondering, a bit of a technical, and maybe I'm the only one interested in this, but with the hair, with the 2- and 4-year-olds, did you have issues across the different sites, and were the reliabilities affected sort of within site? Because, maybe not because of that, you can't maybe say, but did you have any issues with that, just practically speaking?
Shall I answer that?
Yeah, go for it if you want, yeah.
So we're actually still looking into that. We know that we also have some data, from instance, from other projects in South Africa, and we know that people have like these cornrows where it might be a bit more tricky and difficult to get their brain response, because also we're using kind of gel in these tubes, in these metally bits, to get like a good connection with the scalp, because here is otherwise there would just be air, but we need something to like connect.
You can sort of just gel around potentially.
Yeah, so yeah, I think that there's just gel around, but actually that's one of the other things that we're going to be looking at, because so far we've only looked at the UK data, but the next step is then to compare it to the data in India and also to the data in the Gambia.
Cool. And just one follow-up. So I saw your EEG traces looked so nice. And when we did the same with 8 electrodes that were all on the top, we actually had issues with the data quality of sort of these two, because there's a bit of like a thicker bone there, so we didn't really get a good read. But you didn't seem to have that issue with the more posterior positions, did you? Did you find any difference in data quality between the different electrodes?
Not really, but these traces were like to the ear clip re-referenced, and we know that the data is pretty good quality. I think sometimes it can also be the cap fit that is like on the posterior electrodes is a bit more wide. So just like put the cap down. Yeah, I guess it differs, but with this Synovio system, I think it's pretty good.
Thank you very much.
Yeah, just to add as well that I think the cap fit is a big one, just kind of generally like there's like a one shape fits all sort of thing, and obviously people have different shaped heads, and that's natural. And so yeah, just kind of during testing sometimes the caps you're trying to kind of pull them as much as you can, but they have to be comfortable for the children too, and sometimes there's just it's that I would say that's the hardest thing for getting the good signal, is getting the cap like the best fit you can, basically. But yeah.
And there are still kids you don't want to fiddle around too much with the sensors, because you want them to still kind of like you and look at the screen.
Can I ask you some questions?
Sure.
Please, and thank you for your presentation. The first one was is the duration of the experiment, the collection, because I think that you didn't talk about it, the time that you needed to do the collection.
Yeah, you're right, we didn't. My bad. So it was kind of variable, particularly because we used the eye tracking, so children kind of paced it themselves. The first part, so the images and the videos, to memory was average about 35 minutes long. And so yeah, I mean, sometimes it felt very long, these are quite young children. I guess the one kind of benefit of the portable system is you can leave children capped and they can take a little break if they need. Obviously it's not ideal, but they can take a little break if they need to. You can just stop recording and come back again, or you can just pause it and they can kind of have a play if they need to. But yeah, by memory about 35 minutes long for that part, and then the auditory part I think was about 12 minutes, but they tended to like that bit because they could watch something like a Peppa Pig or whatever, or they could play with bubbles, so that was more fun for them.
Okay, and regarding the gel in the caps, how did you do it? You get the gel before applying the cap, or
not in the UK. In the UK, we kind of put the cap on with the metal electrodes already in, and then there's a little hole in those electrodes.
Okay, and you
use a little syringe. Yeah, yeah. Okay. So through that little hole, a tiny little syringe to put the gel in. I think Rianne, correct me if I'm wrong. I think in either India or the Gambia, they did, maybe the Gambia with infants, they maybe pre-gelled and gelled them first and then put it on. We did that sometimes. But I think because, as someone asked earlier about the hair, I think because toddlers, like 2.5 to 4 year olds tend to have more hair, applying it once the cap's on tended to get kind of a better signal, because you could kind of wiggle the hair a tiny little bit to get it through. But it very much, I know other people are trying different things, and I think it depends on children's tolerance. And we did it sometimes pre-gelled if we had a child that really hated it being gelled once it was on the head. Kind of we're a little bit adaptable if we needed to be.
Yeah, that was the other question that I wanted to ask you, is about the children that refused to use the cap. In what circumstances that happen, and what did you try to do to keep the children involved? And you decided there's no way.
Yeah, good question. Again, it's so variable, and I kind of took parents lead quite a bit on, and the children. So I found some of the children really wanted to know exactly what was going on and would ask, what are you doing? What's that? And you could explain it to them in a child-friendly way. This is, we explained the gel, like, oh, this is a bit like hair gel, like some of the mums were like, oh, daddy wears this, something a bit like this in their hair, and let them touch it, let them have, some of them wanted to have it on their finger and feel what it felt like before they were happy with us putting it in their hair. Others, it was like, just keep them distracted and just do it and they'll be fine. So basically very variable, and had to take it by individual child and just kind of let it, took it from parents sometimes. But I mean, parents were brilliant, and there was some children who refused it, but, you know, parents were like, we try, and then they'd be like, okay, but the parent would be like, try again. But there's only so many times you can try, and if the child is very unhappy, you know, is saying no and doesn't want it, we kind of have to kind of take that and be like, okay, fair enough. But yeah, I mean, parents are really, you know, we did things like we put it on ourselves and demonstrated and showed them, and different things like that. One thing I did wonder, kind of, if I did it again, would maybe be to make a child-friendly, like, result, like a story almost of, you're going to come here and this is the cap, and show them, you know, this is like a bit like a swimming cap, and kind of just so parents could actually show children before they came in, this is what's going to happen. Whether it would help or not, I don't know, but it was an idea that I thought in the future might be worth kind of trying, just so kids aren't too overwhelmed when they come in. But yeah, basically very variable.
But sorry, Ellie, I know I didn't run the study, but just to add to it, I mean, you saw out of the 60 odd participants, there were only 2 children who didn't want it. So most kids, and same with babies, are absolutely fine. Like, even if the parent will say they have an aversion to hats, it's always worth having a try. But at the end of the day, we are child and baby led, so we'd never do anything that they're unhappy with, or that is causing upset. But I think generally with the techniques Ellie was saying, either sort of really explaining everything and letting them touch it and show it to them, or if they're not interested, just using distraction, you generally can get the cap on, and then it's sort of up to your stimulus. So if what you're asking them to look at or do is interesting enough, it's much easier to forget that you've got, you know, you've got something on your head. Whereas if maybe something more tricky to watch, it's much easier to remember that, hey, what is this? I don't like it. Yeah, and just another thing that, just to add as well, the ear clip, some again, when some of them were bored, they'd be like, oh yeah, I've got this ear clip on, and then, or it would feel uncomfortable. Sometimes we just kind of stopped, switched it to the other ear, and then carried on, and that was fine. It was just that they remembered it was there now, or it started to bother them a bit, so we switched ears, and then that was fine again. But yeah, there's lots of tricks and techniques, and lots of different things to try. It's the way to do it. Any other questions? Can I just ask a quick one then? In terms of, like, sorry, I know that it was really all about reliability and do these measures work, but the actual stimulus, was that telling you anything about development? And was it different? Like, obviously, were the images, like particular faces that were shown to children in the UK, the same as what you showed to the kids in the Gambia or India? I mean, were there any developmental trends you could actually tell from that, or? No.
We haven't actually looked at that yet. I mean, the age group that we have is pretty small. It's only, there will be like a month in between, and I don't think you will be seeing that much development yet. You kind of need a few months in between. But we do know, for instance, that when children get older, for instance, the latency, like the speed is quicker, and the responses get a bit smaller. That has to do with brain development and myelination in the brain. And so those are like the general things that we see in the ERPs. And there are also differences in responses for faces up versus inverted, for instance. In adults, there's a stronger response, more negative for inverted faces compared to upright. But in infants and younger children, actually, this is the other way around. So then we can also look at that, how it develops across age. But now that we know that these measures are moderately reliable, we can focus on these different ones and then look at how it develops across age with different age points.
Brilliant. Sorry to cut you off. I think Zoom is about to end on us. About 30 seconds left. So thank you everyone for joining us. We really appreciate having people interested in our research. We've got one more coffee talk next week, which will be all about an online study that one of our students ran about action sequencing and action planning. So do join us if you can. But for now, Ellie, Rianne, thank you so much for telling us about Braintools. That was really interesting and very much appreciated. So thanks, you guys.
Thank you.
Thank you.
Thanks, everyone. Bye.